So this diagram shows how the RAS gets activated in Ras MAP kinase pathway. So RAS is an intracellular monomeric GTPase switch protein. RAS is an intracellular monomeric GTPase switch protein. So at first, signals from the outside attached with the receptor tyrosine kinase. So upon binding these receptors undergoes dimerization as well as autophosphorylation. So the binding of ligand causes dimerization and autophosphorylation of this tyrosine kinase. Okay. So this is the dimeric receptor. Then after dimerization, GRB2 and SOS bind with this tyrosine receptor and RAS. So this GRB and SOS, GRB is an adapter protein and SOS is a guanine nucleotide exchange factor. They help to link RAS, inactive RAS with the receptor. Okay. So at that stage, at inactive stage, GDP is linked with the RAS. So upon binding with SOS and GRB2, SOS actually promotes the dissociation of GDP from RAS and binding of GTP. Okay, so after the binding of GTP, this RAS gets activated. So at first, after the binding of external hormone or any ligand to the receptor can cause its dimerization and autophosphorylation. At first, this monomer forms dimer, then phosphate group attached to it by the process of autophosphorylation. After dimerization and autophosphorylation, GRB2, which is an adapter protein, and SOS, which is a guanine nucleotide exchange factor, attach to them, which link the RAS with these receptors, with these dimeric receptors. Then SOS promotes the dissociation of this GDP, which is linked with the inactive RAS. Dissociation of GDP or guanine dinucleotide with GTP. Then after the binding of SOS, it promotes the dissociation of GDP and attachment of GTP. Okay, so after binding with GTP, after the binding of GTP, the RAS gets activated. Okay. So the activation of receptor tyrosine kinase may trigger multiple pathways. For example, activation of EGF receptor activates the RAS dependent pathway as well as RAS independent pathway involving second messengers. The activation of RAS triggered by activated tyrosine kinase are short-lived. So RAS activates a downstream serine threonine phosphorylation cascade that includes MAP kinase. MAP kinase means mitogen activated protein kinase. So activated RAS activate another kinase which is RAF. Then RAF help to activate MEK. MEK means MAP kinase. Okay, then MEK again activate MAP kinase. So after the activation of MAP kinase, it can change protein activity and change in gene expression. So the activation of MAP kinase or the phosphorylation of MAP kinase can phosphorylate many other different protein, including nuclear transcription factor, and mediate other cellular responses. Okay. So the ultimate effect of MAP kinase pathway is a change in the pattern of transcription. Okay. So in the classic MAP kinase pathway, MAP kinase moves into the nucleus where it phosphorylates target transcription factor. In alternative pathway, it phosphorylates a cytoplasmic factor. This may be a transcription factor that then moves to nucleus, or a protein that regulate transcription factor. So this is the ultimate effect of this MAP kinase pathway.